to see about structure of DNA. Coming to the introduction, as we all know that there are two types of nucleic acids, namely deoxyribonucleic acid and ribonucleic acid. Primarily, nucleic acids serve as repositories and transmitters of genetic information. DNA is the hereditary material in humans and almost all other organisms. Most DNA is located in the cell nucleus, but a small amount of DNA can also be found in the mitochondria. It is a polymer of deoxyribonucleotides and is found in chromosomes, mitochondria, and chloroplast. Components of nucleic acids. Nucleic acids are the polymers of nucleotides held by 3' and 5' phosphate bridges. Nucleotides are the fundamental units that make up nucleic acids such as DNA and RNA, and these molecules are essential for storing and transmitting genetic information as well as playing important role in various cellular processes. Functions of nucleotides. Nucleotides are the building blocks of nucleic acids. DNA stores genetic information and acts as a blueprint for the production of proteins. RNA plays various roles in gene expression, including mRNA carrying genetic information from DNA to ribosomes, tRNA serving as the template for protein synthesis, rRNA forming the structural component of ribosomes. And energy carriers, the ATP is the primary energy currency of cells. ATP stores and releases energy through the breaking and reforming its phosphate bonds. This energy is used for cellular processes like muscle contraction, active transport, etc. In cellular signaling, cyclic AMP, that is cyclic adenosine monophosphate, acts as a second messenger in many cellular signaling pathways, transmitting signals from hormones and neurotransmitters. Cyclic guanosine monophosphate involved in various cellular processes including smooth muscle relaxation, platelet aggregation, and vision. In coenzymes, nucleotides acts as essential components of many coenzymes like NAD+, NADP, FAD are involved in redox reactions essential for cellular respiration and photosynthesis. Structure of nucleotides. Nucleotides are composed of nitrogenous base, pentose sugar, and phosphate group. Purine and pyrimidine are the two families of nitrogenous bases that are essential components of nucleic acids like DNA and RNA. They form the rungs of the double helix structure connecting the two strands of DNA. Purine bases are adenine and guanine, which has a double ring structure consisting of a 6-membered ring fused to a 5-membered ring. Cytosine, thymine, and uracil are the pyrimidine bases which has single ring structure. The specific pairing of purines and pyrimidines is crucial for the stability and function of DNA. Adenine always paired with thymine in DNA or uracil in RNA through 2 hydrogen bonds. Guanine always paired with cytosine through 3 hydrogen bonds. This complementary base pairing ensures that the two strands of DNA are held together tightly and that the genetic information is accurately replicated and transmitted. Other than these bases, several minor and unusual bases are often found in DNA and RNA, which includes 5-methyl cytosine, N4 acetyl cytosine, N6-methyl adenine, N6-dimethyl adenine, pseudouracil, etc. It is believed that the unusual bases in nucleic acids will help in the recognition of specific enzymes. The bases such as hypoxanthine, xanthine, and uric acid are present in the free state in these cells. The former two are the intermediate in purine synthesis, while uric acid is the end product of purine degradation. Sugars of nucleic acid. The 5 carbon monosaccharides, that is pentoses, are found in the nucleic acid structure. RNA contains D-ribose while DNA contains D-deoxyribose. Deoxyribose lacks an oxygen atom at the second carbon position. This slight difference in structure contributes to the overall stability and function of DNA. Ribose contain a hydroxyl group at the second carbon position. This difference in the sugar component contributes to the unique properties and functions of DNA and RNA. Nomenclature of nucleotides. The addition of a pentose sugar to base produces a nucleoside. If the sugar is a ribose, then ribonucleosides are formed. Example, adenosine, guanosine, cytidine and uridine are the ribonucleosides of adenine, guanine, cytosine and uracil respectively. If the sugar is a deoxyribose, then deoxyribonucleosides are produced. Coming to the history of DNA discovery, in 1869, Friedrich Mister isolates a microscopic substance from the nuclei of pus cells. He calls this substance nuclein, which later became known as DNA. In 1928, Frederick Griffith discovered a transforming principle that can change the harmless bacteria into disease-causing ones. In 1944, Oswald Avery and Macklin McCarthy identified DNA as the transforming principle. In 1952, Alfred Hershey and Martha Chase confirmed that DNA, not protein, is the genetic material of viruses. In 1953, James Watson and Francis Crick proposed the double helix structure of DNA based on X-ray diffraction images by Rosalind Franklin and Maurice Wilkins. Structure of DNA. DNA is a polymer of deoxyribonucleotides. It is composed of monomeric units, namely deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, and deoxythymidine monophosphate. Chargaff rules discovered by Erwin Chargaff in the late 1940s are fundamental to our understanding of DNA structure. They state that equivalence of purines and pyrimidines. In any double-strand DNA molecule, the amount of guanine is always equal to the amount of cytosine, and the amount of adenine is always equal to the amount of thymine. Species specificity. The relative amount of ATGC in DNA vary between different species. The significance of Chargaff rules provided crucial evidence that supported the Watson-Crick model of DNA structure. The double helical structure of DNA was proposed by James Watson and Francis Crick in 1953 and got Nobel Prize in 1962. The illustration of DNA structure is considered as a milestone in the era of modern biology. The structure of DNA double helix is comparable to a twisted ladder. The DNA is a right-handed double helix. It consists of two polydeoxyribonucleotide chains twisted around each other on a common axis. The two strands are anti-parallel, that is, one strand runs in 5' to 3' direction, while the other in 3' to 5' direction. The width of a double helix is 20 angstrom. Each turn of the helix is 34 angstrom, with 10 pairs of nucleotides, each pair placed at a distance of about 3.4 angstrom. Each strand of DNA has a hydrophilic deoxyribose phosphate backbone on the outside of the molecule, while the hydrophobic bases are stacked inside. The two polynucleotide chains are not identical but complementary to each other due to base pairing. The two strands are held together by hydrogen bonds formed by complementary base pairs. The AT pair has 2 hydrogen bonds, and while GC pair has 3 hydrogen bonds. The GC is stronger by about 50% than AT base pairs. The hydrogen bonds are formed between a purine and pyrimidine bases only. The possible pairs are adenine, thymine, thymine, adenine, guanine, cytosine, cytosine and guanine. The complementary base pairing in DNA helix proves Chargaff rules. The content of adenine equals to that of thymine and guanine equal to that of cytosine. The genetic information resides on one of the two strands known as template strand or sense strand. The opposite strand is anti-sense strand. The double helix has wide major groove and narrow minor grooves along the phosphodiester backbone. Proteins interact with the DNA at these grooves without disrupting the base pairs and double helix. Conformations of DNA double helix. DNA exists in 6 forms: A, B, C, D, E and Z DNA. Among this, bDNA, aDNA and zDNA are very important. The B form of DNA double helix described by Watson and Crick is the most predominant form under physiological conditions. Each turn of the B form has 10 base pairs spanning a distance of 3.4 nanometer. The width of double helix is 2 nanometer. The A form is also a right-handed helix. It contains 11 base pairs per turn. There is a tilting of the base pairs by 20 degrees away from the central axis. The Z form is a left-handed helix and contains 12 base pairs per turn. The polynucleotide strands of DNA move somewhat zigzag fashion and hence named as zDNA. It is believed that transition between different helical forms of DNA plays a significant role in regulating gene expression. Thank you for listening to the video.